Dr. Woeller here for Integrated Medicine Academy. This is the monthly installment for the Great Plains Laboratory complimentary webinars. And in this particular lecture, we're going to go through how toxins adversely affect the methylation cycle. And the reality is this is a pretty heavy-duty talk. Very interesting. I'm going to try and draw correlations from a number of different areas. What I want you to come away with from this talk is a deeper appreciation for how toxicity in many different ways could affect methylation. That's not to say that I am going to touch on every single aspect of the methylation cycle, of genetics, of every toxin, or perhaps every single biochemical mechanism that might exist. So we're gonna cover a number of things. So again, just a good overview. My recommendation for this talk is to just sit back and listen, and to not try and decipher perhaps everything or every slide that you see, but just to get a broad overview, to just at least listen to the information and get a deeper appreciation for how things can link up and where things can impact, because there's a lot here. There's about 60 slides we're going to go through. This entire talk could actually become an entire weekend of talks given through a seminar. There's that much information just in this particular topic. So let's talk about the methylation cycle in general. And when I think of the methylation cycle, I don't think of it in isolation. Okay, it's a cycle, it's a system of biochemistry that's linked to other biochemical systems in the body. We can put it on paper and a pretty graphics like they have here on the slide, and you realize they're really interconnecting cycles or wheels. And the activity of these different wheels, whether it's the methylation, folate, the urea cycle, what's called the BH4 cycle, etc., the activity occurs between the wheels. Okay, how these things interrelate and interact with each other. Methylation, from a simplistic standpoint, is the process of transferring a methyl group. A methyl group is a carbon and 3 hydrogen atoms. So that group being transferred to another chemical to modify its function. Now, in some cases, that could be to turn on or increase the activity of the corresponding chemical, or it could be to turn it off or nullify its effects. The process of methylation, it has a very broad effects throughout the body. It helps with memory, retention and recall. It helps with language development. It helps with environmental awareness. It helps in a tremendous amount of neurochemical mechanisms within the brain and nervous system. It is involved in immune function. It's also involved in detoxification. And some of the research into methylation defects has been going on for years. In one community of individuals is autism. They often have methylation defects, but they're not the only ones. Other neurological diseases like Alzheimer's and other conditions can also be linked to problems in methylation. This is a very simple diagram of the methylation cycle. So we've got methionine. If we think about a clock face, like I think of like a grandfather clock. So we've got the clock, and then we have the second hand, that large tail-like, whatever they call that thing that swings back and forth at the bottom of the grandfather clock. So at our 6 o'clock position on the clock face is homocysteine. At our 12 o'clock position is methionine. And we're moving in a clockwise direction. So we move from homocysteine to methionine, and then back down from methionine to homocysteine. The second hand, or the tail-like structure that swings off the grandfather clock, is called our transsulfuration pathway. That's what basically supports the production of glutathione. So really, this particular system can be separated into two different things. We've got methylation, and then we've got what's called transsulfuration. We'll get more into that here shortly. And then there's two routes that we can convert homocysteine to methionine. There's a route through methionine synthase, and there's a route through what's called TMG to DMG conversion. We'll talk about those here shortly as well. Now, a lot of focus is placed on the MTHFR gene, and there's a number of them, or the enzyme, I should say, the MTHFR, and it stands for methylene tetrahydrofolate reductase. The bottom line is that this enzyme, or these group of enzymes, modify different chemical reactions specifically with regards to folate metabolism, but they can also be involved in dopamine and serotonin, tryptophan metabolism, ammonia metabolism, etc. And so people are running out and they're doing this test just to look at a defect in the MTHFR gene without understanding that it's part of a much broader system of biochemistry. It's not that it's unimportant, but I think at times there's sometimes too much emphasis placed on one specific enzyme. And if you look at this from a broader context, we now can see that our wheels interconnect as they did before, but we can start to get a little bit deeper about what other things they influence. So the folate system, for example, is linked to something called the biopterin cycle. Well, that has its hands in serotonin metabolism, dopamine metabolism, adrenaline, etc. Folate cycle is linked to the methylation cycle. The methylation cycle is linked to the folate cycle. If we have a problem in the MTHFR, it's certainly going to impact the way folate functions. But there are other factors that are at play too, and how some of these other biochemical systems can compensate for certain defects. Clearly, MTHFR problems in a greater context being linked to the folate and methylation cycle have been linked to a wide variety of health problems. Everything from Alzheimer's disease to autism to increased risk for colon cancer and breast cancer to leukemia, cardiovascular illnesses can all have some correlation back to problems in these systems. If we look at autism, they know that many of the autistic individuals tend to have a defect in the MTHFR. Now, the one form of MTHFR that gets studied, tends to get most of the press, is the 677 variety. And the most common variant is the C677T variant. That seems to be the one that most individuals have with autism. It seems to be the one that's most prevalent. And remember, the C677T, its primary function is to modify, or I shouldn't say modify, it's involved in folate metabolism within the folate cycle. Now, some of these defects in these enzymes from a genetic standpoint, they know approximately what percentage decrease of function that enzyme might have when there is some type of genetic imbalance. So for example, we've got what's called heterozygous mutations, and that's where we carry a plus-minus configuration. When it comes to the MTHFR, the best approximately, these are approximate percentages, it reduces the function of the enzyme about 25%, maybe 30%. Okay, so there's going to be some inherent drop in function. That doesn't mean that the enzyme is completely 100% turned off. It's just a drop in percentage. If we've got a homozygous, a plus mutation, we're looking at about maybe a 50% reduction. Now that can be significant. That's not a benign event, but again, you have to take it in context of what else the body is doing and what other interlinking systems are part of this equation. And one thing to also keep in mind is it's not known the exact percentage alteration of function for some of the other enzymes involved in methylation or beyond. But what we do know is that, let me bring this up here, we all have some genetic susceptibility to problems. I mean, if you've gone out and done a 23andMe test, you're going to have certain things within your genetic makeup where there's going to be heterozygous or maybe homozygous mutations in certain things that could increase your risk for a problem. It doesn't mean it's automatically going to happen or that it's even happening. But they're all, potential increases. So we all have genetic susceptibility. So what's the other variable? Okay. Clearly there's problems that may have individuals that may have more problems than others, like the MTHFR or some of the other issues. And so their genetics may be a little bit more vulnerable, but what's the other factor that so many of us are exposed to? Well, it's toxicity. It's the food that we eat. Okay, it's the things that we're exposed to within our home, within the environment. It's compounds in the food. It could be mental emotional stressors, alcohol consumption, pharmaceutical drugs, pollutants, smog, chemicals, mold toxins. I mean, the list is huge. Heavy metals. So these have a significant impact on our system too, and they can sometimes worsen an underlying genetic problem. So it's not always just about throwing more supplements at somebody when you see some of these genetic imbalances. It's also understanding that exposure to different toxins can have a negative impact too, clearly. And there's been a lot of research done into the different aspects of the methylation cycle with a number of different health disorders. Autism has been, the research for methylation problems has been around a long time in the world of autism. One of the leaders in this is Dr. Jill James, who had done a number of studies many years ago looking at impairment in the methylation biochemistry in autistic individuals, showing that these impairments increase the propensity for oxidative stress in children with autism, and then certain defects or problems within the methylation cycle in general that could impair language, could impair awareness, could impair cognitive abilities, could lead to behavioral issues as well. And any time that we create a scenario where there's an increased potential for oxidative stress, that can cause cellular damage within the body, whether it's whether it's in the immune system, perhaps in the liver, the kidney, perhaps it's in the brain or nervous system. These reactions can be systemic. Alzheimer's disease. If you look at the research on methylation problems in autism, it almost mirrors that of what's been looked at in Alzheimer's. If you look at the oxidative stress reaction research in autism, it almost mirrors that in Alzheimer's. Now, I'm not making the case that autistic kids have Alzheimer's or that Alzheimer's individuals have autism, but there is a similarity in the way the body reacts and processes things that it's exposed to. Nor am I making the case that there's an increased risk for Alzheimer's in autistic individuals. I mean, that I'm not making that case at all, but I'm just showing that there are relationships, biochemistry, but the underlying problems sometimes manifest in different ways. Clearly with autism, we're seeing that relationship in younger kids, and Alzheimer's isn't something that's typically happening until much later in life. Let's come back now to this methylation diagram, because again, a lot of emphasis is placed on the MTHFR, but sometimes what gets lost in the shuffle is the understanding of the relationship of this other enzyme called methionine synthase. Methionine synthase is directly linked to the methylation cycle for the conversion of homocysteine to methionine. That is obviously a very important reaction. Methyl B12, the supplement, the vitamin methyl B12, has its function within the methionine synthase complex. You'll notice that there are two pathways to convert homocysteine to methionine. We have what's called the short pathway that takes us through trimethylglycine to dimethylglycine, and we have what's called the long pathway, okay, that goes through the methionine synthase enzyme. The question is why is there two? Well, there's a backup mechanism, obviously, that Mother Nature has implemented here because this reaction is very important. But it's the other piece of the equation that comes around on this side, okay? And this cycle right here is our folate cycle, okay? This is where folate is happening. So there is a relationship between the folate cycle and the methylation cycle. Let's look a little bit deeper at methionine synthase. Here we're back to our broader view. Here's our methylation cycle on the right. Here's our folate cycle in the middle and right. And that bridge in between those two is methionine synthase, which is the MS. Another researcher by the name of Richard Deeth has been around a long time. He's done a tremendous amount of research into oxidative stress reactions in autism, and he came out with this paper many, many years ago that talked about how toxins deplete glutathione, the cellular antioxidant that's so important in our bodies, that it can inhibit folate and vitamin B12 dependent methionine synthase activity. And he specifically listed things such as ethanol, arsenic, lead, mercury, aluminum, and the vaccine preservative Thimerosal. Here's a little bit more of a simplistic image of the methylation folate cycle. So here's our folate cycle on the right, methylation cycle on the right, folate on the left. And one of the things that he mentioned was ethanol, okay, having an adverse effect on methionine synthase activity. So that means that ethanol, alcohol, could have a negative impact on the methylation cycle. So that's one chemical that we know that can cause a problem. Well, it turns out that ethanol can be produced in a lot of different ways. We certainly can intake ethanol or alcohol by consuming it. And what Dr. D showed was that it can alter the function of methionine synthase. There's another chemical that often gets produced in the presence of ethanol, and that is something called acetaldehyde. And acetaldehyde is also something that gets produced when there is a fungal overgrowth of yeast. Okay, we also can produce ethanol because of the yeast overgrowth. So we can not only get acetaldehyde elevations, but we can also get increased ethanol or alcohol production by the natural metabolism of yeast cells that could possibly affect the activity of methionine synthase. One of the things that I've seen over the years with many of the autistic kids I work with is sometimes they don't respond as positively to some of the methylating supplements that we would use, like methyl B12, for example, if they also have corresponding overgrowth of candida, and they're very sensitive to it. It's a very individualized thing. It's difficult to say just looking at a test who is and who is not going to get benefit from a specific therapy sometimes, like methyl B12. That's why a lot of times we're left with just trying things to see what works. But at times, I've seen some kids who are very sensitive to the presence of yeast having enough of an interference effect on the methyl B12 that we don't get the full benefit. I've seen plenty of other situations where kids who had yeast and the methyl B12 works fantastic. So again, it really sometimes just comes down to trying different things to see how that one individual person is going to react. But you can see here where biochemically some of this can fit. The other thing that's known to affect methionine synthase can be heavy metals. This was just one particular slide that Dr. Deeth showed that shows that Thimerosal, the mercury preservative that used to be found in a lot of vaccines, is still found in some, like certain flu vaccines, can also inhibit the function of methylcobalamin. And what it does is it tends to rob the body of this chemical called GSH. GSH stands for glutathione. We need adequate glutathione in order to convert certain forms of B12 in order to naturally produce methyl B12 that has its function within the methionine synthase enzyme complex. Well, there's other heavy metals that can interfere with methionine synthase as well. And as we can see, if we interfere with methionine synthase, you're going to interfere with the ability to convert homocysteine to methionine, which is the sequence that's needed for methylation biochemistry in general. If we can't make that conversion, we've got problems, because too much homocysteine can be toxic to the brain and nervous system. It can also be toxic to the cardiovascular system. So what Dr. Deeth had shown was that mercury, antimony, arsenic, lead, other heavy metals can have a toxic effect on methionine synthase. But we have to think about how these chemicals, whether it's ethanol, heavy metals, etc., one of the things that they do naturally in the body is that they increase oxidative stress. And the function involved in oxidative stress is often either caused by inflammation or itself could trigger inflammation. So inflammation and oxidative stress from a larger context could impact methionine synthase activity as well. And remember, the methionine synthase, again, it's that bridge, right? It is the bridge between the methylation cycle and the folate cycle. There's another enzyme that's involved in methylation. This is the one that's called the short route. And this is where we convert TMG, also called betaine, and we convert it to DMG. And that's done so through an enzyme right here, number 6, okay, called betaine homocysteine methyltransferase. Betaine homocysteine methyltransferase is a very highly zinc-dependent enzyme. If there is marked zinc deficiencies in the body, it's possible that this enzyme doesn't function. But the other thing that I want to point out is that this enzyme is also vulnerable to toxicity. It has a high affinity for heavy metals like mercury, like arsenic, like antimony, etc. The same things that were negatively impacting the methionine synthase activity. Well, now let's expand this out and talk a little bit about the methylation cycle, mitochondria, and then the cellular energy currency called ATP. What's the relationship there? Mitochondria are these organelles that live within our cells. They're like the cell's organs, for example. The mitochondria is what produces the bulk of this chemical called adenosine triphosphate, ATP, And this is the main cellular energy currency that cells of the body use, okay? It's involved in cells of the brain and nervous system, cells of the digestive system, cells of the immune system, the cardiovascular system, all over. Mitochondria are also involved in because of this, aspect, is they're very much involved in cell growth, cell cycling, cell signaling, etc. So they're critically important. And different organ systems have different concentrations of mitochondria. So if we adversely affect mitochondria, we could potentially adversely affect other organ systems throughout the body. And what I'm going to show you is that you can also adversely affect methylation cycle. So within the mitochondria, so now we're looking at a mitochondria kind of in sort of an expanded view. It has two different membranes. It's got an outer membrane and an inner membrane. Inside the matrix, inside the substance, if you will, of the mitochondria is where our Krebs cycle is. Krebs cycle is also called the citric acid cycle. And we actually produce some ATP through the Krebs cycle as things are moving through the Krebs cycle. So if we bring in fat or protein, for example, it gets spun through these biochemical pathways and we kick out ATP. That's a terrible ATP right there. But you get the point, okay? It produces ATP. The Krebs cycle speaks to another system in the mitochondria called the respiratory chain. These two systems are linked to one another. And it's in this system that we produce the bulk of the energy currency of ATP. Various toxins can affect the function at various levels, okay? We can have toxins that affect the Krebs cycle. So it's known that heavy metals can do that. Chemicals can do that. Clostridia bacteria toxins can affect Krebs cycle. Yeast toxins can affect it too. And many of these same toxins can also affect the respiratory chain. So the bottom line is the mitochondria are very vulnerable to toxicity in many forms. We'll talk about what are some things that protect the mitochondria here shortly. There are different regions of the methylation cycle that are dependent on mitochondrial activity. And the biggest one really is, and there's a number of them, we're just going to focus on a few, is this region here is the conversion of methionine to something called SAMe. ATP produced by mitochondrial activity is added to methionine. Okay, once that homocysteine has been converted to methionine, it's added to methionine to become what's called SAMe, or acetylmethionine. If there is a lack of ATP production, okay, via some type of mitochondrial dysfunction being caused by toxins of various forms, that impact alone is enough to compromise and slow down the function of the methylation cycle. Okay, if we come back to this slide, we're now sitting right here at our 12 o'clock position on our clock face. Methionine is at our 6 o'clock. So we've now moved up, okay, from homocysteine to methionine. This system now wants to move back down to become homocysteine again. But as it does so, the SAMe here, this is called SAMe, S-adenosylmethionine, this reaction, this enzyme right here, to make that chemical reaction happen, requires ATP from our mitochondria. And as we move down, we have a tremendous amount of influence on neurotransmitter function, function of the nervous system, DNA processing, the production of RNA, which is important for protein development in the body that has an impact globally on the body, like the immune system, for example. So there's a lot of implications if we start to slow down the function of the methylation cycle. And that could occur here with some kind of blockage because of a lack of ATP being linked to some type of mitochondrial problem, being linked potentially, all this is potential, to some type of chemical or toxic exposure. I need to say more than just chemical, but toxic exposure. Well, what about the folate cycle? Where does that come into play in this? Well, here we come back to this larger view. We've got our methylation cycle on the right and our folate cycle in the middle. We know that these things are linked through the methionine synthase enzyme complex. The methionine synthase can be affected by metals, chemicals, inflammation, oxidative stress. Well, it turns out in order to get adequate folate, or the normal level of active folate, I should say, into the brain and nervous system, it has to be transported in. Okay? There's a transport mechanism that makes that happen. And again, remember, the folate is important because it's interacting as well with the methylation cycle through that methionine synthase enzyme complex. There's a condition called cerebral folate deficiency, which can occur in some children, even some children with autism. And it's known to occur and lead to a lot of neurological problems. It's also been associated with psychiatric problems too. Now, the most common cause of cerebral folate deficiency is an autoimmune process where somebody's developing antibody reactions to the folate receptor or the folate transporter. And a lot of times that can happen because of a dairy sensitivity. But this process is also energy dependent. It's dependent on adequate supplies of ATP that are coming from mitochondria production. It also turns out that in addition to ethanol's effect on the methionine synthase, okay, whether it's ethanol that you're drinking from an alcoholic beverage or ethanol that's coming about from yeast production, it too can have an adverse effect on the folate cycle. So we not only have an impact of ethanol on the methylation cycle, it can also impact the folate cycle too. Let's talk a little bit about organophosphates in something called PON1. Okay, so this is a different looking slide here. If we just kind of orient ourselves to this. So they're just kind of switching this around. Here's our folate cycle, okay? Here's our methionine synthase and our B12. Here's our methylation cycle, right? So here's our methionine at 12 o'clock. Here's our homocysteine at 6 o'clock. And then here's our short cycle, right? Our TMG that we talked about that gets converted to DMG, okay? But I want to focus right here. And this is an important reaction called homocysteine thiolactone, and specifically this enzyme called paraoxonase. Organophosphates, dimethylphosphate, diethylphosphate, are the major metabolites of many organophosphate pesticides. Now, these things are sprayed all over the environment, or can be. People who live in agricultural areas, live near golf courses, live near public parks where they spray. A lot of people can be exposed. These things are really problematic and very toxic and potentially problematic for a wide variety of people. Paraoxonase, or PON, is an enzyme where its structure was figured out based on its link to the metabolism of a specific organophosphate called parathione. And if you do the Great Plains Laboratory toxicity panel, they can measure a chemical called diethyl phosphate that can give some insight into the activity of peroxonase 1. Here's an entirely different viewpoint of the methylation cycle. But let me just kind of orient you again, because even though it's different looking, it's still the same thing. Here's our methionine at 12 o'clock. Here's our homocysteine at 6 o'clock. Here's our short cycle, okay, betaine to DMG or TMG to DMG, so we kind of know that. And then we've got our, well, they're not listing it, but it's our methionine synthase is basically in here, okay? Now notice that homocysteine gets impacted upon by PON1. Homocysteine clearly gets converted to methionine, but homocysteine is also impacted by PON1. One of the other ways that homocysteine goes is homocysteine becomes cystathionine, and that goes off to support the production of glutathione. So there's a lot of ways that homocysteine can go. So you can almost think of homocysteine as a bridge, if you will, between the production of glutathione and the methylation cycle. PON1 is involved in a number of different chemical reactions. Okay, here's our PON1, our peroxonase 1. Okay, I'm just going to put PON. Homocysteine, again, remember sitting at our 6 o'clock position on our clock face, it spontaneously, some of it anyway, can spontaneously convert over to a compound called homocysteine thiolactone. Too much homocysteine thiolactone is toxic in the body. It's not only neurotoxic, but it can be carcinogenic. This is likely one of the reasons of some of the toxic effects of too much homocysteine, is because it's its own toxin. So what does the body try to do with a known toxin? It tries to neutralize it and get rid of it. Well, the body ends up linking it up with the amino acid lysine to basically try and render the homocysteine thiolactone non-functional. Okay, so that's a normal reaction. But the body will also try to reconvert homocysteine thiolactone back over to homocysteine, and it does so through the activity of PON, of the peroxonase enzyme. But also, peroxonase enzyme is responsible for metabolizing organophosphates, these pesticides. So it's got a number of different things that it's tackling, that it's battling. What happens if you increase the activity or the presence of organophosphates? You can overstrain the PON1's activity, right? So now all of a sudden, its function, I shouldn't say its function starts to drop, it's just that it can only handle so much. And we start to get a potential increase in homocysteine thiolactone that has its own toxic effects. All of this, I mean, in so many different ways, because toxins themselves have an inflammatory component, an oxidative component. Those can have systemic effects back into the methylation cycle that could affect the methylation cycle in general and a lot of other areas of the body too. If you were to have some type of inherent defect in your PON1, which some people have, well, then you're going to have an inherent difficulty already dealing with homocysteine thiolactone or any presence of organophosphates. You could take somebody that has no defect in their PON1 and you could totally burden their body with too many organophosphates, and you're going to overwhelm their system, and they're too going to have methylation problems. It just is more, I shouldn't say worse, but I guess there's just a greater propensity for problems if there's also some kind of genetic problem in the PON1. So in a much broader view now, if we link up both of these systems, and so this is a little bit more of a complicated picture, but it's really similar to the one I had showed earlier in this lecture, in that we've got homocysteine at 6 o'clock, we've got methionine at 12 o'clock. Here is our methylation cycle. Here is our folate cycle, okay? And they're both combined or linked because of the methionine synthase. We know that the methionine synthase is susceptible to oxidative stress, inflammation, heavy metals, and that oxidative stress and inflammation can also come about because of endogenously produced toxins as well as other chemical toxins. We know that our ability to transfer folate into the brain and nervous system requires ATP. Any kind of mitochondrial defect could compromise the ability to uptake folate appropriately. If we've got ATP problems, or mitochondrial problems, I should say, we could compromise the back end of the methylation cycle, which then has negative influences downstream. These systems can really be separated into what's called methylation and then transsulfuration. Remember, homocysteine is our bridge. So depending on need, it can go in multiple directions. One of the areas it will go to when the body needs extra antioxidants to maintain or help neutralize against toxicity is it gets pulled down to become glutathione. Okay. Now, there are people that have defects in the ability to make glutathione because from genetic standpoints, or you can run into a glutathione deficit because of being exposed to multiple different types of toxins. The more demand of toxicity you put on the body, the more demand you're making on the body to try to keep up with the glutathione, and then eventually some people start to run into deficit positions. One of the other functions of glutathione that's critically important is it protects mitochondria. So if we have glutathione deficiencies, we're at tremendous risk for mitochondrial dysfunction because the mitochondria can become oxidized and damaged because of internal toxins that naturally exist within the cell through its own metabolism, or things that the body is being exposed to that can also adversely affect mitochondria. And there's other nutrients, vitamin C and catalase and vitamin E and these other types of antioxidants too, but the mitochondria play a significant role. Okay, so the mitochondria really can take a hit and be affected by so many different things: heavy metals, toxic chemicals, inflammation, candida, clostridia, nutritional deficiencies. I mean, the reality is I could have taken this slide and I could have put methionine synthase here in this slide, okay? I could have put folate in this slide. Trying to think, I could have put a lot of things in this slide because they all can, all of these factors could potentially impact on their function. There's a couple of things I want to just focus on before we're done. And a couple of the chemicals. We'll talk a little bit here about mycotoxins, and then we're going to talk about something called NAPG. First, let's focus on mycotoxins. Mold produces mycotoxins. Mold, along with yeast, fall into the biological group called fungi. Candida is a yeast. Aspergillus penicillium are molds, but they're all fungi. And what's particularly troublesome, particularly about mold, is its propensity to produce mycotoxins. And mycotoxins are chemicals, or I should say toxic chemicals, that are produced by a wide variety of molds. And they can have tremendous toxic effects in the body. And unlike bacterial toxins, that these mycotoxins aren't proteins. They don't typically get detected by the immune system, but they can have a broad range of toxic effects. They can disrupt cellular processing of protein. They can affect DNA and RNA function. They can cause mitochondrial damage. They can cause depletion of glutathione. And when we cause mitochondrial damage, we affect essentially the ability to produce ATP. Here's just an example of a specific type of mycotoxin called aflatoxin. Aflatoxin is produced by the mold called Aspergillus. The P450 enzyme system is in our liver. And there are certain individuals that have, some genetic defects in their P450 system. But basically, that system is involved in the first phase of liver detoxification, where we're converting lipid soluble compounds like chemicals and attempting to move them into a position to become more water soluble so that we can eliminate them from the body more easily through the stool. All right, so a number of things can interact with that function. We can also have problems in the ability to produce or maintain glutathione, and the more toxins that we have from various sources will also impact on glutathione. One of the things that's involved in liver function, not only at the cellular level, but within the liver's function or the liver's ability to detoxify, is glutathione itself works within the different phases of liver detox. So as we compromise liver function just because of the presence of a mycotoxin, it can lead to toxicity. And when we have toxicity, what are we getting? We're getting an increased production potentially of inflammation and oxidative stress. And just correlate that backwards to everything that we've talked about in this presentation of where that might affect certain things within the methylation cycle. Certain molds produce certain mycotoxins that fall into a group called trichothecenes. And these trichothecenes are extremely problematic because they too will stimulate the immune system to produce different inflammatory compounds causing immune suppression, tissue injury, oxidative stress. Certain trichothecenes, for example, come from Stachybotrys mold, which is the black mold. Aflatoxin and many of these other mycotoxins produced by mold will affect different areas within the Krebs cycle and then again impact mitochondrial function. So, and in one particular case, if we look at this particular slide, this marker called pyroglutamic is a marker off Great Plains Laboratory organic acids test. And it indicates a glutathione deficiency when that marker is elevated. So if pyroglutamic is elevated, it's indicating a deficiency of glutathione. It can be elevated because of chemicals. It can be elevated because of heavy metals. It can be elevated because of too much fungal, too much bacterial, too much Clostridia toxin coming from the gut. It can also be elevated because of mold toxins. Let me just finish up on just a brief discussion of acetaminophen, something that's commonly used, still widely available here in the United States from any drugstore. And some people have a real problem with it. There can definitely be some toxic reactions to the overuse of acetaminophen. Paracetamol is, it's acetaminophen. It's what they call it overseas. A lot of times people who, instead of getting Tylenol, they get what's called paracetamol. It's the same thing. And paracetamol, Tylenol, acetaminophen breaks down into a chemical called NAPQI. Notice that we need glutathione to neutralize it. It converts NAPK to a non-toxic metabolite. I'll go through slowly on this slide. Here's our acetaminophen. Okay. Some of it gets converted naturally through something called phenol sulfur transferase and then excreted in the urine. If there's a defect in our phenol sulfur transferase enzyme, that could compromise our ability to metabolize acetaminophen. Sometimes people who have high levels of oxalates can have a compromised phenol transferase system. We can spontaneously form NAPK by taking acetaminophen. But if we have plenty of glutathione around, it'll essentially neutralize it, create a non-toxic compound that we'll get rid of. Okay, so basically we should have the ability, we ideally should have the ability to neutralize it as much as possible. Well, you can see where this is going is that doesn't always happen. Okay. A large amount of acetaminophen can overwhelm the system. You can get too much NAPK formation that can have liver toxicity. It can also damage other sulfur-containing proteins throughout the body, including the brain and nervous system. It can, over time, lead to a depletion of glutathione. And as we deplete glutathione, our mitochondria become more vulnerable, our ability to detoxify in the liver becomes compromised, and it sets up a whole cascade of events of inflammation and oxidative stress. Let me go back real quick. Okay, so that's one of the downsides clearly of acetaminophen. Well, some additional research really had shown that, again, acetaminophen can decrease the synthesis, i.e. the production of ATP at the mitochondrial level, and that acetaminophen can deplete glutathione, or the pool of glutathione that exists. That's clearly not good. And in doing so, that can damage the mitochondria. And then it takes us all the way back to the methylation cycle. Okay? Because again, we need adequate ATP from mitochondrial activity in order to create the ongoing process of methylation through the ongoing conversion of methionine to this SAMe chemical. And if we don't, the methylation system becomes sluggish. Downstream from that, the transsulfuration pathway becomes sluggish. Our ability to maintain glutathione becomes problematic, et cetera, et cetera, et cetera. Okay, so that's the core information here. So these are complicated systems. We can't just look at things in isolation. You don't want to just do an MTHFR test and that be it. Okay, to really get at the core issue of complicated, chronic, complex health disorders, we have to, as practitioners, start to broaden our view and learn how to integrate different types of testing to be able to make those correlations about what are the things that are triggering problems and what are certain things that aren't. We can't assume everything. We have to many times confirm it through laboratory assessment. The Great Plains Laboratory GPL Tox Profile is a fabulous test. It gives you an indication of exposure to a wide variety of chemicals. I do a lot of this test in my practice, and there's many other practitioners doing this too. But it needs to be taken in context with other testing that is done as well, at least ideally. Great Plains Laboratory has something called the EnviroTox panel. And this is the combination of the GPL tox with something called glyphosate, which is a very toxic herbicide, along with the organic acids test. And I've done webinars on this for Great Plains, talking about every single one of these different tests. And I actually teach an entire one-day seminar on how to incorporate the organic acids test into practice. So my preference, if at all possible, is to do these series of tests to get a much broader viewpoint of what true toxicity is really occurring and all about. In addition, to incorporate the mycotox testing as well, because the mycotox testing, looking at mold toxins, is another component of toxicity that some people don't even know they have, that could be causing tremendous issues in the body. And as you can see in the title of this presentation, there is ways to correlate and understand how all of these different toxins, including heavy metals as well, can impact the methylation cycle. So again, it's not always just about, throwing supplements or, just depending on one specific therapy to try to get the job done. It's taking a broader view and trying to make these correlations so that you can come up with a much more comprehensive program. The GPL mycotox test is also an incredibly important profile to run too. So we're looking at, what Great Plains is looking at is 7 different mycotoxins that are occurring from the presence of 4 different molds, even certain toxins that are produced by the Stachybotrys mold. Okay, so all of those are available through Great Plains Laboratory. If you have the ability to order those, I'd highly recommend it. We also have a website called Lab Test Plus, labtestplus.com. You can go to that website and look at the different menu of labs that are available. Many labs from Great Plains are held on that website too. Any lab that is ordered through Lab Test Plus will come with what's called a written interpretation of the pertinent findings, and then some action step suggestions based on that information when warranted. So you can go to labtestplus.com and get more information. If you have questions, you can email to labtestplus@gmail.com. I also have a course that's a very comprehensive course. This course is specifically designed for healthcare practitioners called Toxicity Mastery. It's 9 modules. Each module lecture approximately, an hour and a half to 2 hours. And there's many breakout lectures also in this course. And it's all about understanding the GPL tox test, the organic acids test, glyphosate, heavy metal testing, mold, mycotoxin toxicity, and different types of detoxification therapies, from heavy metal chelation to sauna therapy to ion cleanse to glutathione, bowel and liver cleansing, etc. So you can go to toxicitymasterycourse.com and get more information. You can get access to the course material at any time. So you can join a course if it's currently going on live, or if you want to join when the course is not being given live and still have access to the recordings, that is always available. We're currently in the process of doing a live version of this course. In fact, we just did our first module last week, so there's 8 more to go. That's going to extend throughout the summer. If you have any questions, you can email to toxicitymasterycourse@gmail.com, and this is also opened to non-healthcare practitioners as well. That's totally fine. I just want people to understand that the content is developed for healthcare practitioners, but anybody is open to taking it. If you'd like to get access to this special report booklet, you can text the word toxicity to 66866, and you'll get a copy of this environmental and chemical toxicity book. And that's also an overview of the Toxicity Mastery course. And then I'm always available for private consultations, do my private practice. The best email to reach us is scmedicalcenter@gmail.com. So I appreciate everybody's attention today. If you submitted questions, Great Plains will usually they'll email those to me in a few days afterwards. So there's always that option. But yeah, if you're interested in any of the courses that we have, please check them out. If you have any questions about things, you can always email my practice at the SC Medical Center at gmail.com. And if you're interested in getting access to some of the lab testing on your own, you can check out the labtestplus.com website. Okay, so this is Dr. Kurt Woeller for the Great Plains Laboratory complimentary webinars. Thanks so much, and we'll see you next month. Take care.